So good evening, and I'm really sorry for the technical hitch that many of you may have experienced with joining us this evening, but I'm really glad and hope that you're now able to see and hear me. Let me start by welcoming you and thanking you for joining us for this Research to Reality webinar. For those of you on this webinar who don't know who I am, my name is Sheridan Gould, and I'm very proud and very privileged to be the Executive Director of Weitzman UK. I'm hoping that we're joined by our friends and supporters from around the world, and thank you very much for being with us today. And thank you to my colleagues in Rehovot for coordinating this online event, but I would ask them next time around to make sure that the joining procedure is a little easier than it's been so far. So the wonders of technology, that's I hope is allowing us now to listen in today, but really I hope very, very much that this is perhaps one of the last of our online events this year, and that hopefully we will have the opportunity to be together and meet in person. And certainly with these technological hitches that we're having, that really is a great hope. So we're truly, truly honoured to be joined today by Professor Jacob Hanna, who is going to talk to us about growing organs outside of the body. I'm going to leave it to you to decide if what Jacob is going to tell us is indeed science fiction or fact. But before we hear from Jacob, I'll shortly hand over to the chair of Weitzman UK, Dr. Arabella Duffield, who will do a formal introduction. But I'd like to say a few personal remarks about him. Firstly, I'd like to say, like all of his Weitzman colleagues, Jacob is a truly remarkable scientist. And I recall when he first joined the Institute as a young scientist, quite some time ago now, the former president of the Weitzman Institute, Professor Daniel Zeifman, used to come and talk to all the executive directors around the world about the various young scientists that were joining and how excited he was about them all, and how it was very important for us to help fundraise to ensure that they could establish their labs as young scientists. But in Jacob's case, Daniel was very, very excited about him and said his research was really, truly outstanding and very special. But he also said that he was going to be very expensive, that the work that he was planning to do required extremely expensive equipment, and that it would be a real challenge for us fundraisers to make sure that we could secure the necessary funding to enable him to do his work. Well, I'm really pleased to say that two very special friends of the Weitzman Institute, Ilana and Pascal Mantoux, were extremely quick off the mark and very quickly agreed to support this young scientist. And I have to say that they really made a fantastic investment, and they should be super proud of all that he's achieved. And I believe they continue to support him today. Weitzman UK, in a slightly more modest way, have also supported Jacob in his work through his Making Connections grant, a programme that's very dear to Weitzman UK, in which we're very keen to ensure collaborations between UK scientists and their colleagues in Israel. And Jacob was one of the early recipients of our Making Connections grants. In 2008, he received a grant from Professor Roger Pedersen and Dr. Mark Cotter at the University of Cambridge. And I know that that grant, even though it was a modest one, has established some fantastic collaboration. And I know that even though they're not being funded anymore by us, they continue to stay friends. Jacob's also received grants from the BIRAX program here in the UK. And I've even seen his research referred to on a BBC television show. So he really is a superstar scientist. But we're not here this evening to listen to me. I will shortly hand over to Arabella, but before I hand over to Arabella, we have a very, very brief little video that we'd like to share with you about all that is limitless and wonderful about the Weizmann Institute of Science. So let's listen to the little video and then we'll hear from Jacob. Thank you.
Let's imagine a world with no science at all, where thousands of years of discovery simply never happen. How then would we explain our world, expose its mechanisms, actions, and origins? And how would we create all the technology that we take for granted, hardware, software, healthcare? Because none of this would exist, of course. And how would we understand that most powerful device, the human brain? How it works, and why does it sometimes not work? Who would we turn to for answers to the many challenges that face mankind? And that starry sky, what's up there? Aren't you curious? The questions are limitless. So aren't you glad that there is a place where people are asking them? Scientific and technological breakthroughs only exist because of basic scientific research. And at the forefront of global basic research is the Weizmann Institute of Science. For 70 years, Weizmann researchers have been asking questions most of us can't even imagine. Their curiosity led to the creation of Israel's first computer, one of the first in the world. It was a Weizmann scientist who worked on the algorithm that makes your online transactions secure today, discovered the life-changing multiple sclerosis drug, Copaxone, and created new cancer-fighting treatments. And today, Weizmann scientists are creating DNA-based computers so tiny, 1 trillion can fit in a drop of water, using quantum electronics to build supercomputers, developing intelligent materials inspired by nature, and exploring the limitless potential of machine learning and artificial intelligence. They are finding clues to our changing climate and protecting our planet's resources for the future. They are combating disease with powerful diagnostic tools, drugs, and therapies, and they are teaching the next generation to dream, to think, and discover. Looking back, we owe the greatest milestones of human achievement to our curiosity. Each of us must do all that we can to ensure that the trajectory and pace of discovery never falters. If we want the best of human existence to be ahead of us, we need basic scientific research to take us there.
Hello, everybody. My name is Arabella Duffield, and I am the chair of Weizmann UK. Thank you for joining us. I'm very sorry about the technical hitch earlier. As Sheridan said, hopefully this will be one of our last times on Zoom. I'm very excited that Professor Prof Hanna is talking to us tonight. As some of you may recall, he was here in London about 6 years ago when we heard about his promising stem cell research. And we're now going to hear about the amazing breakthroughs that he and his team, and indeed his collaborators from across the world, have achieved. Jacob started his academic career at the Hebrew University of Jerusalem, where he earned his BSc in medical science, his PhD in microbiology and immunology, and an MD in clinical medicine. He conducted his postdoc research at the Whitehead Institute for Biomedical Research at MIT and joined us in 2011. He's earned really many awards and accolades for his pioneering research. I'm not going to bore you with all of them, although of course it wouldn't be boring, Jacob, I'm sorry, but I won't tell you about all of them, but I am proud to say that he did get the Sir Charles Claw Prize. In 2014, he was elected as a member of the Israel Young Academy and was also featured amongst the under 40 innovative scientists by the journal Cell. Last year, he was announced the winner of Prospect magazine's Top Thinkers of 2021. And I guess at the end of this presentation, we'll all know why. Enough from all of us. I just want to say one thing. I will be chairing the Q&A after Jacob's talk. So if any of you have any questions, please do put them in the chat. Thank you very much. Over to you.
Okay. Good evening, everyone. Thank you, Sheridan. Thank you, Arabella, for inviting me. And thank you, everyone, for the interest in science and hearing about my work and what we're trying to do at the Weizmann Institute. So allow me now just to share my presentation with you. Okay. Okay. So I can you see, I hope everyone can now see my first slide of the presentation. So my lab has been focusing on studying stem cells and early stem cells for the last 11 years, and that's what I started my postdoc in as well. But I think, you know, the big question and the challenge starts with a big challenge that we face in modern society is how do we come up with body part replacements, whether you're thinking about patients who need organ transplant, tissue regeneration, or whether actually you could think of people who are healthy, they want to have better joint, better muscles. So it's not about also being sick, but also about being healthy. And the challenge is really, where do we come up with such a source and an easy source for such organs for transplantation, and overcome the problems of tissue matching? As you know, it's very hard to find donors. Even if you find a donor, the DNA is never matching, so that you will always have some kind of rejection, and patients have to be put on immunotherapy, and deal with rejection and so forth. So that's a big problem. And that's basically where the stem cell field started to become more and more interesting. The stem cell field, as I try to convince you, starts and ends with early developmental biology. In the end, we're all facing this amazing question: how are we all, as mammalians, all start from a single cell after fertilization to make an entire embryo? So this is a big question that scientists have been trying to address for years. But what was fascinating, that scientists originally in the UK about 40 years ago showed that if you take a very early embryo in the mouse origin called the blastocyst and dissociate it in a dish, you can grow what is called embryonic stem cells. So these cells that are very, very early stage, that are undifferentiated, can be isolated from early embryos in mice. And then, of course, as this was shown in humans and many other species. And these cells can, if you put them back into the embryo, they can make an entire embryo. So they still keep this pluripotency. They can give rise to every cell type. And that's why people thought that, oh, this is a very promising source to use them for the starting material to more organs for transplantation. So I guess that's what's really gave a lot of attention and a justified boost for the stem cell field. However, the field has progressed a lot, and there were a lot of ethical and technical issues with using embryonic stem cells from embryos. So embryonic stem cells, as I told you, they are taken from embryos that were donated for research, which basically usually couples who underwent IVF therapy. After many years, the frozen embryos, they can be discarded, or the parents agree to donate them for research. And that's how those ES lines were derived. However, a problem, if you think with the embryonic stem cells, if you think, let's say I'm a patient and I need a transplant of a liver, the DNA of these stem cells that are donated from an embryo is not identical to my DNA. So I might have a source of cells for transplantation, but we did not solve the problem of rejection. And many years of research actually has led to ask, can we really make stem cells without using embryos or gametes? Or as you can see here in this image, if you take the patient, can we take a skin cell or a drop of blood and go directly back in the dish to this embryonic stem cell state? And 15 years ago, a scientist in Japan, Yamanaka, got the Nobel Prize for discovering that he can take 4 proteins that are usually expressed in the egg, and he imposes them on differentiated cells, and about 1% of these cells go back to become stem cells. And these are called iPS cells, or induced stem cells. And now we're talking about cells that are identical genetically to the donor. This can be done in mice, this can be done in humans, regardless of the age of the donor. And that was very exciting. And just to show you the potential of this technology, one of the studies that we showed early on in this field is to really show that we can make this promise of stem cell therapy. Or for example, we took mice that had a genetic disease called sickle cell anemia. It's a mutation that causes the red blood cells to be fragile and become sickled. And we took skin fibroblasts from the tail. We made iPS cells in the dish, and these iPS cells are genetically identical to the donor mice. And then actually, we corrected the mutations in these iPS cells. So we don't want to give back 6 cells with a mutation. So we can correct mutations in iPS cells. We can make bone marrow, and then we transplanted bone marrow back to the same mice, and we get about 80% cure of this disease in mice. So that's just to demonstrate the power of all of this in vitro technology of making stem cells and use them for transplantation. And since then, a lot of research has been done, but we still face a major challenge. So if we're talking about stem cell therapies, there are 2 parts for this. We want to take a skin cell back to become a stem cell. And then we do not, and it's dangerous to inject undifferentiated stem cells. We have to move the cell forward and then transplant it back to the patient. And you can think in our body, there are about 250 cell types, and there are many labs that are specializing and working. How do we move forward? How do we make kidney cells? How do we make insulin producing cells to really specialize and make mature or pure populations that can be used for transplantation? I would say actually it is surprising because it was thought that the challenge is going to be actually how do we go backwards? But that actually was solved quicker, and we can make iPS cells efficiently and safely very well today. But what has proved very difficult actually is going forward. So when we're trying to take these stem cells in the dish, we can make very few cell types nowadays that are, as I said, authentically equivalent to those found in the embryo. And this is a major challenge. And we're trying to understand, if you think, what are we trying to differentiate? So what we need to do is to mimic how the embryo makes its organ. The embryo does it best. So we want to understand the principle how the embryo makes its organs, learn these principles, and impose them and apply them in the petri dish. But this is a problem because we have what is called the uterine barrier. Mammalians, whether it's mice or humans, mostly develop inside the uterus. So we can grow embryos until the blastocyst stage, which is 64 cells, very early on, both in mouse and humans, outside the uterus. And this is, of course, the basis of all IVF therapy. However, we have to put back these cells back into the uterus. And what is immediately as the embryo implants inside the uterus, you start having this very rapid drama of what's called gastrulation and organogenesis. So very quickly, let's say in the mouse, within 4 days, the embryo starts to organize and make all its organs. So this is all happening inside the uterus that is non-transparent, that we cannot control the environment, and we cannot see exactly and rapidly what is happening. So if you think of the mouse that has 25% pregnancy, 20-day pregnancy, 75% of it we just has to happen inside the uterus, and we have very limited access to it. And these processes of gastrulation, so gastrulation is basically the budding of the organs, and then you have organogenesis, are very exciting also for us as scientists. And a very famous British developmental biologist actually had a famous quote saying that it's not birth, marriage, or death that are truly the most important day in your life, but it's actually gastrulation. If you gastrulate correctly, you're going to be healthy of birth defects. So that's why there is a lot of importance to study these processes. And as I mentioned to you again, we can grow very early embryo until the blastocyst stage, and we can put it even back into a uterus in a surrogate mother and make an entire mouse. But all these later stages where you can see the embryo moves from being a ball of stem cells into quickly an elongated structure that quickly makes all its organs. And you can see by day 11, everybody can tell that this is an embryo. But as I said, we are unable to grow mouse embryos or any embryos at these post-implantation stages. We cannot even move them from one uterus to the others. So we're very limited. We cannot make perturbation, let's say a mutation, and follow the outcome. We cannot control the environment. What are the nutrients really surrounding these embryos? And these are all parameters we wanted to study. And if you look at, basically, there were very few attempts dating about 100 years ago only to make embryos grow outside the uterus. Most successful one, again, is from the UK, from Dennis New in the 60s. But again, he was able to grow them for about a day and a half with very low efficiency, and they were mostly abnormal. So overall, they were there seemed to be a glass ceiling that couldn't be broken. And 7 years ago, as a side project, a very talented team of 2 PhD students from Mexico, a staff scientist, Rada Masarwa, and a bioinformatician, Noah Noverstein, and my team decided, well, can we revisit this with the new tools we have? Can we at least grow them 1 or 2 extra days outside the uterus? So our approach was to think, okay, let's try and design a machine. Let's think of this some kind of a ventilation machine. We don't want to ventilate the embryo lungs. We want to ventilate the surroundings of the embryo. And slowly we started sketching different parts of what this device, electronic device, will be. And what are the sensors, what are the valves that are needed. And this took us 7 years of building and iterations to really improve this machine. And as you can see, the final product looks not very nice because it looks like something we make in our own garage, and this is what we do, but it works. And what you see here, it's an electronic device that can control a lot of parameters: temperature, oxygen, CO2, atmospheric pressure, and other parameters. And we can connect these. On the left, you can see this gas regulation module connected to an incubator on the side. And this incubator is special because it has a wheel, because we've learned that these embryos, in order to grow, we need them to be moving inside this wheel. So we came up with this apparatus, and you can see what we had to develop as well, the media. So you can see that fluid, the yellow fluid. So this is the fluid in which the embryos will be growing. We don't have a maternal blood supply, so we have to give them the nutrients they need. And you can see here in each bottle of glass, you can see an embryo between the size of 0.5 centimeter or 1 centimeter growing inside this apparatus. So you can see after many years of study that we can grow mouse embryos, and you can see when the system is working, each bottle has an embryo spinning in the wheel. And you can see on the left, when we're starting with day 7 embryos. So it's just a ball of stem cells. And then look, 1 day later, it has a yolk sac developing. You can see the heartbeat. 1 day later, you can see how the embryo has a head and legs. 3 days, you can see the embryo not only a heartbeat, but it has red blood cells. So it has its own blood system. And these embryos grow for 4 days until day 11 of development, which marks really the completion of making all the organs of the embryo. And we did this very, very efficiently. And just to show you again, we had to really make up all of the techniques of how do we handle these embryos? How do we feed them? How do we move them? So you can see here how this is when the system is working, but there was a whole kind of tech development of how do we change the liquid, change the food for these mammalian embryos and move them every day. And so we take them out, put fresh media, fresh food, and put them back in the system, and they are very, very happy embryos. So we did a lot of testing to really show, are there any delays? Are there any defects? And as you can see here in this example, actually, we did not find differences between in utero, the controls, what is normal for the embryo to grow, and the embryos grown ex utero. And the embryos were normal. Everything was expressed in the right place and the right time, and there was no developmental delay. But actually, we were then quite greedy. Because as I told you, we were starting with day 7, and day 7 is already the process of gastrulation has already started. And we wanted to make a point that we can make the entire process of gastrulation or organogenesis outside the uterus. And really, as a proof of principle, there was this open question, in a mammalian, can we capture the entire gastrulation process and organogenesis outside the uterus? So we wanted to start earlier for day 5 or day 6, and this took us 2 extra years, but my students were very persistent and they managed. Now they do this in a static conditions in regular plates. They take the same media, and we can grow embryos from day 5 for 2 to 3 days very, very efficiently outside the uterus. And you can see really the power of being able to watch a mammalian embryo develop. So these embryos, for example, are labeled in red, and we're starting now at day 5, which is a ball of cells. And watch how the embryo now starts to growing. Also the yolk sac and the placenta are growing. And then you're going to see this kind of arrow moving, the red arrow, which becomes basically the spine moving, defining the head and the legs and the other side of the embryo. So again, this is another example. You can see how the embryo is growing very dramatically, the yolk sac around it and the head, and it starts taking shape. So we can really see now embryogenesis in front of our side, outside the uterus, and it is normal embryogenesis. And in the end, we could combine all these protocols together, and we can grow embryos from day 5 and day 11, which is about 6 days, which is about 1/3 of the entire mouse pregnancy outside the uterus. And the emphasis is not only about the number of days, but is the phases we capture, that we can really get organogenesis and gastrulation in a mammalian outside the uterus. So what is this starting to bring us to think of moving, study how an embryo moves from being a ball of stem cells to an fully organized embryo. So you can see how not only there's a change in the shape of the embryo, but a great increase in size, about 20-fold. And what these experiments really tell us, that the embryo has the capability to self-organize. The uterus is basically feeding it and nurturing it, which is very important, but it's not the one patterning the embryo. And this is conceptually very, very important to prove. So that was exciting for us, but as I told you, we want to make a research tool. So we don't just want to grow embryos, we want to be able to play with them and manipulate them and ask, can we change them and follow the outcome over time? So I'll show you a couple of tools we developed. For example, we can take an early embryo and quickly infect it with a virus that can label or cause a mutation for the embryo and follow it over time. So you can see here now these embryos, we label them with a green color, and you can see within 1 day, the entire embryo is labeled, and it keeps growing outside the uterus for 5 days. It is normal without delay. So we have now a tool to really perturb these embryo and follow the outcome over time. We also wanted to look, can we really look in high resolution how certain organs mature? So if you look here, we're looking at a mouse embryo, and this is the brain, and this is what's called the neural tube, and the brain has not closed yet. And some of you may know, the folic acid is very important in pregnancy, really to prevent defects in this process. And we can really video live imaging and show how the brain closes, just like you can see this zipper movement, to really watch how normal brain development can occur normally outside the uterus. So this was, we're very excited to be pursuing this project, and we think this is the start of a very long path for us, because there are a lot of challenges that we're trying to answer. One of them is actually, can we grow embryos for longer periods of time? We are not seeking to grow in the entire pregnancy, but at least until all organs are formed, and this is an important question. Can we use this to look at other animal models that beyond the mouse? For example, the rabbit is very useful because the rabbit embryo shape in the early on is actually very similar to the human embryo, much more similar than mouse is to human. And we're now starting to adapt this system to be able to grow rabbit embryos so that they can be another model that we can use it by extrapolation to learn about human embryos. And finally, now we're starting to probe this novel concept of what is called synthetic embryos. So all what I've showed you, we are actually growing naturally formed embryos. These are embryos that were formed after a natural fertilization. We can take them out at day 3, at day 5, and then grow them. But there was a big question in the field that is still open. Can we actually just take these stem cells that we grow in the dish and aggregate them in 3D, and can we grow an embryo out of them? And this is called synthetic embryology, because we're only doing this from stem cells and we're not using an embryo. However, the major problem was, if you couldn't grow a natural embryo, if you didn't have such conditions, why would you expect to succeed in growing a synthetic embryo? But now that we've shown we can grow natural embryos outside the uterus, this becomes now the positive control, the reference. So we now know these conditions, and we can start asking, can we make some kind of what I call organoembryoids, an embryo-like structure from stem cells outside the uterus that have organs that can be very useful for disease modeling or therapy or transplantation and so on. So these are the future directions for us for the next 10 years. And I would like really to thank a lot of important people and teams that helped us over the last really amazing 11 years for me at the Weizmann Institute. The Weizmann UK, I want to mention the Making Bridges grant. This has really been very helpful, because it's not only about the grant, but about really meeting excellent UK scientists and know them in person. And this really influenced a lot of my research. I'm very thankful to Ilana and Pascal Mantou, a wonderful couple who helped establish my lab and continue to partially support the activities that are done. And you can see them here in the picture, actually even trying to do some experiments on these embryos. I really want to thank what I call the Weizmann family, or my family at the Weizmann. And this really includes the wonderful staff at the Weizmann at all levels, whether it's scientists, whether it's support staff, whether it's Weizmann UK. It's just been a joy to work together and try to promote science and get people excited about what we do. And I want my own family as well that supports me to be lucky and dedicate this time to do research. And with that, I want to thank you all, and we're now open for questions through moderation with Arabella.
Can I just say, Jacob?
Yes.
I have seen so many talks, and that is literally the best scientific talk I have ever seen or heard. It was so clear.
Wow.
And so interesting. I'm really not making that up. I find physics difficult, but that was amazing. Seriously. So thank you very, very much.
Thank you.
And I'm pretty sure there's going to be a million questions. I've got a few that came in earlier. and I'm lucky enough to be allowed to pick which ones I want. You've covered off the bit, I think, about the collaborative research with other organizations and the UK in particular. So I don't think I need to do that one. But I suppose the big question that I'm interested in is, you've spoken about how your research before does sit at the kind of contested boundaries of life. And I just wonder how you manage the ethical issues that come up.
Yeah. So,
and sorry, and how that relates to Israel or not being in Israel, or that's, I think, of interest maybe.
No, definitely. So I would actually start saying that Israel and the UK are very similar overall in the terms of ethical guidelines that they follow. Maybe perhaps we just imitate the UK, but really, yes, this type of research definitely raises ethical questions, ethical challenges. And I have two parts to my answer on this. One is saying that, there are a lot of examples, for example, just because someone can make a nuclear bomb, you should not ban nuclear physics, which is important for space research and energy. Just because someone can make a very dangerous virus, you don't want to block virology research. And we now see how important it is to do viral research. Mm-hmm. So we should tackle on and really discuss the ethical challenges. And of course, when we're talking about the ethical challenges about trying to do this with human embryos, this is the relevant questions. And of course, we're currently not pursuing any experiments with human embryos, but it's important to discuss the way I view this personally. It's not a black or white question. I think in the end, there is an ethical price, but the ask is, what is the benefit versus cost? Mm-hmm. It is important to emphasize that us in the future, or any other scientist who's trying to use human embryos for research, it's important to emphasize that we are not asking couples to go get pregnant or make IVF for the purpose of donating their embryos for research. There are millions of frozen embryos that are not going to be used, and they are getting discarded. And the question, can we actually use them for, let's say, 2 more weeks and learn about this critical person, because there is a great benefit. So my personal view is not that it's, I would say, there's absolutely no ethical issues. I have no difficulty. No, there is a difficulty, but I just think that the benefits are so large that I think there is justified needs of doing some of these experiments in a controlled manner.
Thank you. I mean, I, of course, totally agree with you, but thank you. It's very interesting to hear that. Now, I've just been sent some questions. I'm sorry, I'm going to have to read them. Is that okay, or have you got them? Shall I read them? I'll read them.
Go ahead, yeah.
So the first question I've received is, there's work being undertaken relating to a global problem of type 1 diabetes. ViaCyte and others are using encapsulated stem cells implanted in the pancreas. Yeah. Could you expand on this research?
Yeah, so actually, it's actually been in the news lately. So there are, I think, as I told you, as a field, we can make, we know how to differentiate very few cell types. Some of them are dopamine neurons that are actually the ones that are damaged in Parkinson's. And also there's great progress on making insulin producing cells, which is beta cells. And there are a couple of companies that actually now trying to transplant beta cells. The nice thing actually about beta cells, they don't have to sit inside the pancreas to do their job. They can be just a capsule, under the armpit or that so far. And there was actually lately in the New York Times, there was the first patient who was actually cured of diabetes. So that is very promising. And I want to actually, on the other hand, just really, what is really remarkable, all what I've talked to you is 15 years old. And this is actually very little time. And despite that, this very little time has happened, there's just so much progress. And I think I expect that diabetes and Parkinson's are perhaps one of the early successes, hopefully in the next year. So there are ongoing clinical experiment started this year for Parkinson's in Sloan Kettering.
Okay, thank you. I have another question from the audience, which is related to one that was sent in earlier as well, which the question is, which organs are you focusing on, or which ones do you think are probably the most promising to grow outside the body? I've got here, it says heart, question mark, kidneys, question mark.
Yes. So, you know, as I want to say we still don't know. So I think, you know, sometimes actually as I was thinking today about it, is many times we are subconsciously influenced in our science. And I think one of the great things for me was with the Wiseman UK exchange grant was working with Roger Peterson. So those who don't know, Roger Peterson is a very, very famous stem cell scientist, and he's a developmental embryologist. And as you saw, basically I'm shifting more now to the study of the embryo. And I think he's, you know, these discussions that I had with him have influenced a lot and really pushed us, you know, slowly to work on the embryo. And we are trying basically to make what I call the embryo. So it's kind of what might be a structure that might have all these organs. We don't know which one of them are going to be normal or not. So we're trying in general just make the budding of all the embryos at this point, but we might have to focus later depending on the outcome.
Okay. That's very interesting. Thank you. Well, there's so many questions I have, but I'm trying to let others in. One of the issues someone has asked, and there are two again related questions from the audience, is the speed at which your kind of research, I mean, I imagine, well, anyway, it could be translated into public health benefits that we will see in our lifetime. I'm going to say, you look around the same age as me. If that's rude. I hope not. And, you know, will we be able to afford this kind of research? You know, is it going to be, Sheridan made a comment about it being very expensive, and I imagine it is, you know, scientific research is. So there's a question around that.
I mean, actually, you know, it might, the answer I'm going to give you, it might sound like a lawyer's answer, but that's not my intention at all. But I want to say, if you think today of, for example, there are now treatments with antibodies that, you know, you can get antibody treatments for immune therapy, for cancer. So they've become actually routine. So if you look at the time, you know, what it took to bring antibody treatment to the clinic, it took about 50 years because people had to discover antibodies. Then they had to discover the cells that made them. And then had to discover how to control them. And then the first drugs came out in the 90s, and then they were very, very expensive. But then the technology got improved and competition. And then we have many more antibodies now, and the cost is much better. So I think this is something we see over and over again, how this one, and I think, yes, I mean, as I said, this is still a very young field, but progress is being made. In the beginning, you know, you could think maybe the cost will be high because it's actually really quite cost intensive. Actually, and it's not at the stage actually that the companies are trying to make money. It's just actually it's expensive to do this. But like everything else, I think, you know, that these things get slowly become more routine, more affordable, more efficiently done with time. So I am overall optimistic. Even as I mentioned, even for some of them when we're talking, I do believe that for Parkinson's and diabetes, in the next 10 years, I think this will start to become quite abundantly practiced.
That's amazing. Thank you. If you don't mind, I'm just going to go back to a kind of Weizmann-based question.
Sure.
And ask why you chose the Weizmann when clearly the world was your oyster. Why did you choose us? And do you think it's given you an added advantage, the kind of ethos of the Weizmann, or could it have been anywhere? You know, you could have met the English professor somewhere else, and you know, I'm just curious to know about that.
Yeah, I mean, I think one can never know. I can only say that I'm very happy with the choice I made, and my intuition at the time, which turns out to be very true, that the Weizmann is a place, because perhaps it is a little bit outside, perhaps, you know, geographically, you know, perhaps it's a bit distant. It's a place that allows a lot of freedom thinking, and there is no pressure to follow the herd conceptually when you're pursuing science. And it's the opposite, actually. You're really encouraged to go after really bold ideas and really what you would call weird ideas even. And I felt that, and I was drawn to that. And just as an example, you know, this project started because I received an what's called it's a Weizmann Award called the Kimmel Award, where every year a scientist is given $1 million to do a crazy idea, whatever he wants. And that's how this project started. Otherwise, we wouldn't have that. So really, this is just one example of many. And of course, then, you know, you come to it's an excellent place. It's a very friendly place, a very collaborative place. I think there is a feeling also like, as a group, like we're successful, but we're still successful in doing it against the odds. And I think that kind of brings also a very sense of community and work together. Really everybody is trying to push each other up and make their ideas true. And I felt it back then, and my intuition was correct. It is true. It is what happens.
I'm very pleased to hear your answer on that one. I suppose I have just, I've got 3 more minutes so that we can be on time. I have just one last question that was sent to us before. What do you commented that you're moving more towards the development side of anything, of start looking more into that. But if there's one thing that your lab is working on now that you think will be very exciting in 5 years, could you name it? Do you think what do you think we can expect from you in 5 years? What should we be looking for?
Yeah, I think what we are mainly focusing on is trying to just take these stem cells and put them in these devices that we made. And we are seeing evidence that we get what we call like an artificial embryo-like structure. So it's not an embryo, but these are things that are organized, that are starting to bud organs. And we very much want to analyze and understand, and we think they can be a source of cells for transplantation. So I think that is what we will be focusing on for the next 5 years.
Thank you, Jakob, so much. Can I say it again? That was literally my best talk ever. And I have never said that before. I hope it's being recorded, and I'm just going to hand you over to Sheridan to say goodbye. And thank you.
Okay, well, let me just say thank you all for persevering with the technical hitches at the beginning. I'm sure you'll agree that it was definitely worth the wait. And I think, I'm not sure we should be calling you Professor Hanna. Should we be going into the science fiction realm and calling you Professor Frankenstein? I seriously hope not. But really, yeah, Jacob, you've just demonstrated your patience, your perseverance, and your unbelievable clarity of thinking and outside the box approach, the machinery that you and your students developed to create these embryos and working outside the box in these incredible brains to come up with the idea. I think was just simply fantastic. So thank you so much, and we wish you lots and lots of good luck with the program. We need these embryos. We need these organs to help people who desperately need them. Thank you, Bella, for the Q&A, which was fantastic. Thank you, everyone, for joining us on this Zoom. Let me say that whilst the pandemic is not quite fully behind us, things are moving on in the world, and travel to Israel is now opening up. If any of you would like to arrange a visit to the Weitzman to see for yourself just how remarkable and wonderful it is, do please get in touch. We're always happy to organize visits for our friends and supporters. And as I said at the beginning, I do hope that the next time we will all meet will be in person and not on these boxes on our computer screens. So have a lovely evening or a lovely day wherever you are in the world. Stay safe and well, and keep up this fantastic work, Jacob. It really is remarkable. Thank you.
Thank you all.